Hello everyone. First, I'm going to start introducing myself. I am Ulises Rodriguez and I am PhD student in biomedical science at the National Autonomous University of Mexico. I am interested in molecular biology and bioinformatics. Probably many of you have heard of the cuatro ciénegas Coahuila basin in the north of Mexico in the state of Coahuila, which for a long time has been the study site of the group led by Valeria Sousa until recently in the year 2016 we have begun to study a new pond characterized by the presence of microbial mats with dome-shaped structures that also have a strong smell of sulfur, and additionally interesting physicochemical properties have been noted, as well as a great microbiological diversity. In an initial characterization, it has been reported that this site is hypersaline, since in the wet season in 2016, a salinity of 5.6 practical salinity units and a pH of 9.94 were reported. It's also important to mention that the site remains under saturation of salinity in dry season. Because of the physicochemical properties and the formation of the dome-shaped structures, we have named this pond as archean domes. As we have already mentioned, the properties of this site could suggest that extremophile microorganisms could be proliferating there. Since most of the extremophiles are very difficult to culture in the laboratory, or at least not with the conventional microbiological techniques, a more appropriate approach to study them is through culture-independent techniques, such as metagenomics. In this study, we have used next-generation sequencing to obtain DNA from 22 samples that were taken from the surface to a depth of approximately 50 centimeters. After the samples were obtained and sequenced, we processed the sequencing data using bioinformatics tools. Then, we performed quality control and cleaned up the sequencing reads, and then we assembled the reads to obtain contigs. And after we get the contigs, the following step was to obtain metagenome assembled genomes. This mag assembly procedure is very similar, or at least I imagine it like playing with a giant bag of multicolor Legos, and the goal of this Lego is not to assemble figures, but the impressive thing is to assemble the genome or genomes that are in the bag. To assemble these genomes, we use two different programs. The first one was MaxBin2 and the second one was MetaBat2, and the result of the assembly of mags for each metagenome was the input for the binding refiner program, which is an R tool that tries to remove chimeric genomes. After that we tried to remove the chimeric genomes, we then evaluated the completeness and contamination of these mags, and we tried to classify them by phylogenetic similarity with the GTDB toolkit. And although we obtained a large set of mags, we decided to filter them to continue working only with those mags with the integrity greater than 70% and contamination less than 10%. The results of these mags or this quality filter was 52 mags classified within archaea domain and 277 mags classified within the bacteria domain. Within this set of mags that we are reporting, 4 classified within the phylum of Asgarchiota stand out, which is a phylum that has recently begun to be described, and it has been proposed that they are the closest prokaryotic relatives to eukaryotes. Following with the results, it is also important to note that the mags we obtained from the archean domes are distributed throughout the entire archaea phylogenetic tree. The same thing happens with the 277 mags classified within the domain of bacteria. They are distributed throughout almost the entire phylogenetic tree of bacteria. Then we have started to perform the functional characterization of mags through the search for Keg orthologs, and there are genes related to methane metabolism in archaea outside the Urarchaeota phylum. However, although there are genes related to methanogenesis, important genes such as MCRA are not present in the MAX classified within the order Thoraciales of the Asgard superphylum. The members of Asgard superphylum are very difficult to cultivate with conventional microbiological techniques, and since only one organism cultivated in syntrophy with other microorganisms has been reported to date, it would be possible to suggest that the archean domes system can serve as a model for the study of the interactions that take place there. It has been reported that in natural anaerobic environments, sulfite and methane generation is ubiquitous, and sulfate-reducing bacteria and methanogenic archaea always coexist. It has been reported that in natural anaerobic environments, sulfite and methane generation is ubiquitous, and sulfate-reducing bacteria and methanogenic archaea always coexist. And it has also been reported that the sulfite pathways build a bridge with methane metabolism through methylcoenzyme M, the most central precursor substance for methane generation. Finally, as a partial conclusion, although we cannot determine this mechanism of methanogenesis, the presence of MAX from the phylum Desulfobacterota could be a good start to further study methanogenesis-related syntrophic relationships in MAX from Asgarchiota members and members of other Archaea superphyla.